Hello, my name is Yakov Davidov, and today I'm going to talk about Designit. It's a flexible engine to generate experimental layouts. And this is teamwork, and none of this would be possible without Juliana, Guido, and Balázs. And first, I'm going to start with a motivating example why we think batch effects matter. So here's an experiment design. You have different treatments in different rows, and you have a concentration gradient in columns. And you have controls in the top and the bottom rows, F and A. And this could look like a good idea initially because you have two sets of controls, right? So you should be able to normalize your data even if you have batch effects. But when you look at actual results, you see this. So there is a gradient at the top row F, and there is no gradient at the bottom row. Sorry, there's a gradient at the top row A, and there is no gradient at the bottom row F. So we don't know why this happened. Maybe a batch effect only affected one row. Maybe it was a pipetting error. Maybe there was a gradient affecting the plate in some way. So at this point, it's basically impossible to normalize your readouts. And the only thing to do is rerun your experiment. And we could have prevented this, right, by distributing controls a bit better on the plate. So in general, we think about our observations as a sum of biological effect, a systematic error, which includes batch effects, and random error, which you usually cannot control. In the worst case scenario, your groups of interest, the groups you want to compare, will be coinciding with your batches like depicted here. And at this point, it's basically impossible to do anything because your batches and your groups are completely confounded. And more generally, we usually have a limited set of samples and we want to have precise group estimates and we want to avoid confounding. And to do this, we need to distribute groups of interest evenly across batches. Two known techniques to create balanced designs are randomization and blocking. So randomization is very simple. You just randomly assign your samples to locations which you have. And unfortunately, it doesn't work too well, especially when the sample sizes are low, because you can have poor grouping by chance, like is shown here, for example, where black mice are only in the batch number 2. And sometimes you will have experimental constraints which will not allow you to go fully random. Blocking, on the other hand, is a more useful approach, especially when the sample size is small. So you first group your experimental units together, and then you try to distribute them evenly, like is depicted here. Since we get this type of requests quite often, we created another package called Designit. And the idea is that we try to avoid batch effects and gradient effects. And to do this, we allocate samples to experimental layouts. And we try to make the more or less general framework, which allows you to customize various aspects of how this is done. The main data structure is called BatchContainer. It's an R6 class. And it stores your experimental dimensions such as batches and plates, sample annotation, and also it stores optimization history. And the function which is the driver behind all of this is called optimize design, and it will optimize assignment of samples to batches, and you can customize how it behaves. The most important thing is the scoring, and then you can change the optimization protocol and how the samples are shuffled. And I will start with a simple example where we have 31 samples and we want to assign them to 3 equally sized batches. First, we need to create a batch container. So we do this by calling the new method. We provide our dimensions, which is batch. So there's 3 batches and 11 locations. And then we randomly assign our data. So this is our sample sheet to the locations in your batch container. Before we start optimization, this is how it looks like. And you can see, even though it was random in this case, it's not ideal. So for example, the second batch is mainly dominated by the first group. And there's basically no fifth group in the second batch. So it will be very difficult to compare the two when this experiment is done. Before we start optimization, I wanted to show you how you get samples from your batch container. You call this get samples method, and you get a table which will have your batch locations here in the red, and then you have your sample information on the right, and in this case means that this location is empty. Now let's try to optimize this. First, we need to think, how do we balance this? So in this example, it's very clear. We want to have treatment group to be even across batches. And maybe we also want to make sure that sex is not very unevenly distributed. So in this case, we have higher priority for the treatment group and lower priority for sex. To tell optimized design later that this is what we want, we create a list of scoring functions. The first one is group. So it's OSAT score, which is essentially a chi-square test-based method. And we tell the function that we need to make sure that group is distributed evenly across batches and the sex is also distributed among batches. There's a list of two different functions. And in this case, the order matters because we will later tell the optimized design function that it should take the first function with higher priority than the second one. And now this is how we call the optimized design. So we override our batch container. We create a new one, right, based on the old one. We provide the list of scoring function. And the most important thing, we provide this acceptance function. So acceptance functions tell optimized design when the solution is better. And we tell it, we use accept leftmost improvement, which means that the scores on the left have higher priority than the scores on the right, which means that if there is improvement in the score on the right, but the left score decreased, like it became worse, we shouldn't accept this change. This is how it works. After optimization, this is how the result looks like. That is probably given the unequal sizes, given that there are 31 samples and 11 samples in each batch, that's probably the best solution you can achieve in terms of group distribution among the three plates. And the sex distribution also looks very good. And when you call plot trace, you could see how the optimization was changed, like how the scoring functions were changing over time. And when this is done, you can again call get samples to get this new table. The batch allocation will stay the same, and you will get new sample assignment, which corresponds to the best solution which was found so far. Now I'm talking about plates. With plates, you have different types of effects, batch effects. So for example, you can have edge effects, like in this example at the top, it's probably related to bed plate sealing in this case. Then you can have gradients. Sometimes it's related to changes in the temperature. It could look something like this. And then sometimes you can have row and column effects, which sometimes are caused by pattern issues, but could be also caused by different things. And what you want to do in case of plate is you want to equally distribute your samples across the two-dimensional plate, right? Then whatever effect you have, it should average out. And to show you an example, here we have a batches which are 24 well plates. And we want to optimize things within the plate and then across plates. And we want to, again, balance things first by treatment group with a higher priority and sex with a lower priority. This is how we do this. We first create a batch container. This is already probably familiar to you. You have dimensions. There are 3 plates. Every plate has 4 rows and 6 columns. And then in this case, we use assign and order, which will basically assign samples in the same order they appear in the table you provide here. And when this is done, you can use the helper function called plot plate. You provide your plate variable, row variable, and column variable, and the color and the title if you want, and this will generate you a plot like this. Sorry. And we see in this case, it's really far from optimal because, for example, the first group is only on the first plate, the fourth group is only on the second plate, and the third plate only has the fifth group. The sex is also not distributed very well, so we want to fix this. We will use a two-step approach. First, we will optimize across plates, as was done before, and then we will optimize within the plate. And the way to do this looks like this. You call a function called optimize multiplate design. You provide your batch container, and then you simply provide the list of across plate variables. And in this case, it's group and sex. And within the plate, it's only the group. And then you provide names of your variables, like plate is plate, row is row, and column is column. And when you do this, you get the following assignment. So you see that The group distribution is very good. It's much better than it was before. And the sex is also well distributed among plates. Within the plate, it's not so well distributed, but we didn't provide this variable here, so this is somewhat expected. You can also plot optimization traces. This is the first iteration, the first step where you were only optimizing across plates, right? So this is the group and the sex score. And then you will have tree optimization, one for each plate. So this is here, I'm showing only the first plate. And you see that the plate number 1, which is red, score is decreasing, which is good. And the plate number 2 and number 3 are not shuffled on this iteration. You will have this in the next 2 iterations. Now, in some cases, this is sufficient and we provide this helper function, but sometimes you want to maybe do this stepwise. You want to see what's happening between the steps. Maybe you want to customize certain things, how things are shuffled. And I will show you how to do this if you don't have this optimized multiplace design wrapper. So we start with the first step, which is optimizing across plates. And we provide a list of scoring function as was done before. There is a group score function and a sex score function. And again, we use accept most improvement. And this should basically be the same as the first example I showed you before the plates. And the second step is a bit more tricky. So we create a new scoring function. It's actually a list of scoring functions, which is generated by MK plate scoring functions. You provide your batch container, your plate row and column, and the group you want to balance. So in this case, because we have 3 plates, it will generate as 3 different scoring functions. And then we can pass it to optimize design, but there is a tricky bit. We already distributed samples among the plate equally. So we don't want to shuffle across plate. We only want to shuffle within plate. To achieve this, we use something called shuffle proposal func argument, which is essentially a function which will suggest to optimize design which things to shuffle around, and then to check the score. If score is better, then accept the change, otherwise go back. So there's a specialized helper function called shuffle with constraints, which will look at your sample table and make sure that conditions for the source location and destination locations are satisfied. So in this case, we only ask that the plate of the destination should be the same as the plate of the source location. And this will make sure that things are only shuffled within a plate. And this is written more or less like you would write in dplyr in the filter function. So this will directly access columns in your table. And I'm showing you this because sometimes you maybe want to customize a couple of more things. So it's good to understand how this is done internally. Otherwise, I would always use the wrapper because it's just less code to write. And now I will give you a glimpse at the more complex application. So we have three treatment conditions. We have 59 animals. They are from 2 different strains. We want to distribute animals to cages, and we want to avoid confounding by sex, weight, and age. And in this case, it's an annual experiment, so it's even more important to make sure that this is done right. We don't want to repeat this unnecessarily. Here is the list of constraints we have, right? There's a number of animals. And for example, some constraints which you wouldn't think about if you're not working with animal experiments is that males from different litters cannot be in the same cage because they could start fighting. And the end goal when you have this animal list and treatment list is to align cages to racks like this. And here every color represents different treatment. And I'm not going to show an example here. You can check vignettes on our website. But when you have such a complex problem like this, it's always good to think about how to split it into simple problems which you can execute sequentially. And please don't be overwhelmed by this example, but I will just give you an idea how to solve this. So you first start by assigning treatment to animals, and you want to balance things like strain, sex, weight, age, and so on. Then when this is done, you allocate animals to cages, and you want to make sure that it's uniform and it's balanced across cages. And when this is done, you arrange cages into recs. Again, ensuring balance of treatment, strain, sex, and so on. And yeah, please check an example on the website if you're curious. To summarize, I think the framework we created is quite general and adaptable. So we can do simple optimizations, but also quite complex ones with multiple steps. You can customize basically everything. You can customize the scoring, the acceptance criteria. For example, we have simulated annealing protocol implemented. And you can also customize how shuffling is done, which is very important. We provide a bunch of helper functions like for plotting and for shuffling and so on. And we provide some vignettes for common use cases. Please also check the function references, maybe find something useful for your case. And with this, I would like to also acknowledge Marta, Sabine, Min, David, Konrad, Solveig, and Fabian, who helped us with feedback, with the presentation, and basically making all this possible. And yeah, please check our package. We have discussions open on our GitHub page. Please let us know what you think. If you have questions, feel free to reach out. And thank you very much. Bye.